Hi, I'm Jenna Beildby, and today I will be presenting our research on the gut microbiota of lean and obese mice on behalf of our research team, which, in addition to myself, includes Austin, Gracie, Emily, and Grayson, and our advisor, Dr. Cooper. With the high-fat, high-sugar Western diet that's common in society today, obesity has been increasing in prevalence. This brings with it a host of other health problems, making obesity a critical field of research. Our focus is specifically on the gut microbiome and its relationship to obesity, as the bacteria living in your gastrointestinal tract play an important role in digestion and management of nutrients, including fats and sugars. Our primary goal is to compare the gut microbiota of lean and obese mice, looking for any potential differences. A long-term application of this could lead to therapy involving manipulation of the gut microbiome. Our secondary, more recent goal is to examine the role of inflammation in relation to the gut microbiome and obesity-linked diseases, as past research has indicated that inflammatory bacteria play a role in diabetes mellitus and cardiovascular disease. We've used a number of mouse cohorts over the years with the same basic principle of a control group and a group with obesity induced by a high sugar diet, though the specific sugar has varied from year to year. Our current data were taken from one cohort with a high sucrose diet and another cohort with two obese groups having high fructose and high glucose diets. All of our research so far has relied on fecal samples. From live mice, we have taken fresh samples and used them to plate bacteria for culturing, both in aerobic and anaerobic conditions, to assess the species diversity in the lean and obese mice. Some samples from live mice will also be used for DNA analysis, although our primary source so far has been from sacrificed mice. We extract the bacterial DNA from the fecal sample, determine the concentration of the DNA in the sample so that we can then run PCR to amplify bacterial DNA if the species for that primer is present. After that, we visualize the results with gel electrophoresis. Our DNA analysis results so far came from a cohort of female mice where obesity was induced by a high sucrose diet. We ran PCR using primers for each of the bacteria listed here on the slide. Our expected results came from articles in the existing base of scientific literature. Some bacteria were not observed at all in our experiments, but whether that's because they weren't present or our primers need refining, that'll need further research. For the results we do have, most aligned with the expected values, except for Bacteroides theta iota omicron, which was present more in obese mice rather than lean mice. The images included here are from some of our gels, where you can see the brighter bands indicating the presence of the bacterial DNA. Our cultures presented here come from the current year's cohort of male mice, with the obese mice in two groups, high fructose and high glucose. With these preliminary results, we're mostly looking at growth and diversity in general. Some trends we noticed included more growth and diversity in the obese mice generally, but especially in the mice on the high fructose diet, and much less growth and diversity in the control mice. Another general trend was that there was overall more growth and diversity in the aerobically cultured plates as compared to those cultured anaerobically. Fungal growth was observed on some of our culture plates, but we're going to need further research to determine whether that's due to contamination or if there actually are fungal species present in the gut of these mice. We've not yet identified any of the bacteria species in our cultures, but intend to do so in the future. One tool that will help us with identifying the bacteria species is this dichotomous key, which we developed as an identification scheme based on biochemical characteristics. It was created using a list of bacteria that we're likely to find in the mice, also from previous articles in the scientific literature. And we're using tests that are all things that we can run here at Corbin. So with that, we aim to use this in the future as we culture more bacteria so that we can build a profile of what bacteria are present in lean and obese mice and note if there's any differences between the two. Looking to the future, as I just mentioned, we would like to continue culturing gut bacteria and use the test in the dichotomous key to identify the species. Again, keeping with our goal of looking for any differences between lean and obese mice. We would also like to continue looking at just overall general diversity between the lean and obese mice, and for that we'd be using some statistical tests. One example would be the Shannon Diversity Index. We also intend to continue with DNA analysis, as we still have samples that we've not yet analyzed. Additionally, we can use DNA analysis alongside the dichotomous key for the bacteria that we're culturing, giving us a two-pronged approach to identifying bacteria species, thus improving the accuracy of our identification. As a more recent goal, we're currently in the process of developing protocols for examining inflammation. At this point, we're considering testing intestinal tissue and markers in the blood, but may try to look at some other methods as well. Thank you for listening.